Thanks very much for the invitation to participate. We very much enjoyed all three of the previous talks, which set things up. I'm going to shift out of the central nervous system to muscle, to the neuromuscular disease, Duchenne muscular dystrophy, and actually talk about some of the challenges, less probably on how it's been used, but some of the challenges in using it in this disease, particularly, especially as we think about things related to genotype specificity. Just a couple of comments. Here's my financial disclosure. I'll mention it later on some data from several of these companies, but I'm not representing any of them here in this context. And to have this conversation, we have to begin with discussion of the dystrophin gene and protein and what it does. It's an enormous gene comprising 79 exons spread over about 2.5, 2.4 megabases of the X chromosome. Those 79 exons encode a 427 kilodalton protein that consists of an actin binding domain, several hinge regions, and a central, all those green marks there actually represent spectrin-like repeats that form something what's often sort of described as a shock absorber for the forces in the muscle fiber as it contracts. Because then at the C-terminus, there's binding to the dystroglycan protein complex. Via that dystroglycan protein complex, stabilization against the extracellular matrix. And there's some additional C-terminal sequence that has binding partners for a variety of proteins. In the absence of dystrophin, the forces of muscle contraction and result in friability of the membrane. And friability of that membrane leads to leakage at the membrane and leakage out primarily of creatine kinase, which is the first biomarker we use diagnostically. Elevated creatine kinase in the range of 50 to 100 times normal is typically what we see first in Duchenne muscular dystrophy. I will return to that as well a little bit later as its use or potential use in the trial setting. But I'll just talk about Duchenne muscular dystrophy, the severe phenotype with absence of dystrophin, really in the days before any of our sort of usual therapies. Typically, children would be off their feet by about 12 years of age. These high elevations of CK were the diagnostic criteria. Becker muscular dystrophy, a classic definition of it is boys who walk longer than that, but actually it's a little muddy. We really kind of have used in many of our studies that look at genetic modifiers where we're splitting up groups, an intermediate group that has ambulation beyond typical Duchenne, but less than the milder forms of Becker muscular dystrophy. And then Becker muscular dystrophy can be loss of ambulation at age 18 years old through age, through long into adulthood, including some symptoms that syndromes that are really associated with only elevated CK by itself or isolated muscle aches and pains with exertion. So really, the dystrophinopathies are a continuum. And as we'll point out, many of the therapies that are being developed are really intended to move a boy from the Duchenne side of the continuum into a more Becker-like side of the continuum. And the way that's done is really, the approaches have really relied on many of them have relied on observations regarding genotype-phenotype correlation. The most mutations in this gene, about 2/3 of them, are deletions of one or more of these exons. There are other small mutations in the gene, point mutations that account for about 1/3, including nonsense mutations in all other categories. But the first therapies were really developed and come out of the idea of these large deletion syndromes, large exon, multi-exon deletions. That observation right after the gene was cloned and identified was defined as the reading frame rule. Unsurprisingly, those mutations that interrupt the reading frame and allow for no expression of a functional dystrophin are those that are associated with Duchenne dystrophy, and those that allow some portion of a functional, some partially functional dystrophin are associated with Becker muscular dystrophy. I won't get into the details of this, but one of our concerns clinically is that what we see on a genomic testing does not always predict the final organization of the messenger RNA, so we have to have this with caution, interpret this with caution, but about 90% of the time this prediction holds up to be true. Now, this observation of in or out of frame led to the first therapies for Duchenne dystrophy, which are exon skipping therapies with antisense oligonucleotides. Those naked antisense oligonucleotides here with the PMO backbone is what the first four were described. The idea is to they mask in the pre-mRNA an individual exon's definition sequence, leading to exclusion of that exon from the mature mRNA. As a result, or there's a restoration of the reading frame. And 4 such compounds have been approved for therapies dating back past for more than a decade now. Actually, all of them have received accelerated approvals. None of these have had confirmatory trials published yet to show to reach full approval. And each of them have been relatively inefficient. I won't go into all the detail here, but I'll point out that, for example, for the first of these approved, eteplirsen, the change was a significant increase in dystrophin measured by a variety of measures, but a statistically significant increase, but still to levels of less than 1% of normal, so not terribly striking amounts. The observation of therapeutic in-frame deletion, though, really derived from the first examples of this, and this led to the development of microdystrophin gene therapies. The observations of patients who have very large internal rod domain deletions, these spectrin repeat element domain deletions, that are associated with ambulation well into the 7th decade, with deletions of about 46% of the protein. And that observation led to the idea of microdystrophin therapy, which I'll return to in a moment. One of the challenges for this, though, is that all internally deleted or all Becker-like mutations, all the dystrophins that are made from those are not equivalent. We can see this in this example from a variety of patients from a large collection, a large database. We have about 2,000 patients and about 400 samples from patients that we look at. And each of the red arrows marks a Becker patient sample. These are all different amounts and different sizes of Becker-like proteins. And you can see one of them is about 15% normal levels, and one of them is about 140% of normal levels on this blot. We've changed our quantification a little bit since we did this, but in this blot, 140% compared to a normal male control. And despite these amounts, the boy with 140% went off his feet in his mid-20s, and the patient with about 15% went off his feet at age 70. That's loss of ambulation, LOA at the bottom. So it just highlights that the amount of an internally deleted dystrophin alone is insufficient to predict outcome. It really is the structure and the functionality of that protein has to be a part of that. Now, obviously, this is relevant to things like microdystrophin gene therapies. Elevidys from Sarepta was approved with accelerated approval in June 2023, accelerated approval for 4 and 5-year-olds. And in last year, in June of 2024, received complete approval for all ambulant patients and accelerated approval for non-ambulant patients. Now, this approval was not without some controversy. The integrated review document at the FDA actually recommended not approving this, and the decisional director memo overrode this for approval. The major concern about it was the fact that it failed to meet its primary endpoint, and the data has subsequently been published in this, what was called the Embark trial. In fact, although the primary endpoint of what's called the North Star Ambulatory Assessment, a measure of function, individual functional measurements that's important to climbing, walking, doing stairs, and other things, failed to meet that endpoint. A variety of other multiple secondary endpoints did all meet significance, really suggesting efficacy. So this was perhaps what part of the controversy. I'll note, though, as well, there's data, in the Embark trial, there's data on dystrophin expression of this microdystrophin isoform. And by Western blot analysis, it reached about 35% of normal levels. One of the other controversial things, perhaps, or confusing things in the integrated review was that there did not seem to be a correlation in individual patients between the amount of dystrophin and how individual patients did, which makes it a little bit challenging to assume that this is a useful biomarker in this disease. So it is approved, and we do prescribe it. I don't mean to cast doubt on efficacy of it. I'm just saying that as a biomarker of disease, this alone may not be sufficient. We'll note that there are, in trials, multiple different isoforms therapeutic isoforms being expressed. These are different versions. The R here represents those spectrin-like repeat elements. I'll just call out solids, SGT, transgene and Regenx RGX's transgene. Data from those trials are going to be presented this weekend. Each of them are different transgenes, though. They are actually functionally different transgenes. The solid transgene incorporates, marked in red, two spectrin-like repeats that are binding sites for nitric oxide synthase, which is predicted and has at least experimentally been shown to alter blood flow at the muscular level, which might be important. And Regenx's version of the transgene incorporates C-terminal versions of the protein as well that are binding sites for a variety of binding partners of dystrophin, suggesting that when we look at amounts and compare amounts between these three transgenes, that will be insufficient by itself necessarily to allow prediction of benefit, because they actually will have different functions. And I'll note here, we'll hear some data from the Chamberlain group later. It's at other sessions of this meeting, as we look forward to future versions of this, sort of second or third generation of dystrophin replacement therapies, make use of a variety of methods, including striking data published just last year on a split N system to reach expression of full-length, mid-size, or full-length dystrophin protein. So the quantification in comparison between these will be difficult. So we again suspect it may not just be the amount, but also the functionality. I want to digress just a little minute to show one example of why we think these kind of concordances in understanding what the protein is important. Here's an example where that 15% patient I showed you there, he actually died, one off his feet at 70 years of age, died at age 77. We found multiple families who carried the same mutation that was responsible for this, which is a nonsense mutation in exon 1. This led to the identification of an alternative translational initiation site within exon 5 of the gene that allowed re-expression of a really highly functional protein, of 413 kilodalton protein. And one of the things we've done is to look at this, we began looking at it in the context of how we could make use of that iris to generate that functional version of the protein. And we made an observation that in patients with duplications of exon 2, the most common version of this, they had DMD, and in fact, they had no iris activity. But patients with deletions of exon 2, they in fact had full iris activity and had no symptoms. And in fact, those patients walk around doing very well until late adulthood. So this suggested if we could think of a way to use skipping of exon 2 as a therapy, we could get wild-type dystrophin, or we could overskip exon 2 and get to a highly functional version of the protein. I don't want to spend too much time on our own data, but I'll just share. We took this to a trial using a vectorized system where we used the U7 small nuclear RNA, and that U7 small nuclear RNA encodes a gene that normally is involved in histone gene splicing, and we repurposed with an antisense sequence directed toward our exon 2. And we made a vector with 4 copies of this in it, and published all the sort of off-target data and so on. And we did a trial, a small trial, funded by a family foundation, actually, in which we had sufficient dosing for 3 patients. One of them was about 9 years of age, one of them was about 13 and a half years of age, and we only had enough left for an infant. So we treated them all at the same dose, and we showed, this is a little hard to see in the back, but you can see the infant is the third group of columns for each of these graphs. And the infant had really robust skipping of exon 2. That black marks the deletion of exon 2. And this was associated with an increased and climbing amount of dystrophin. So in this case, where we get to levels of 80% or more of dystrophin, of a highly functional version of dystrophin, we think it's actually reliable because we've pegged this as a biomarker, because we've pegged this dystrophin to a clinical outcome of patients where we found now a dozen or more patients who express it natively, and we see it at very high levels. I think in the case of dystrophin, where the natural experiment has been done, where we can say this protein gives us this outcome, that's really relevant to using it as a surrogate biomarker. I want to return to CK. CK is a problematic biomarker in many ways. I pointed out that we use it diagnostically. This is from a fairly recent paper from Korea, where they looked at patients and categorized them in these different sort of categories of the ones I described before, of severity of dystrophinopathy. And you can see quite elevated serum CKs at birth that declines by age 3 or so, still in quite high range, 20,000 range for Duchenne patients, but it does decline over time. It declines with loss of muscle mass. It declines as activity slows down. So it's an imperfect biomarker. It's often included in studies, but I should add as well, in both Duchenne patients and Becker patients, it's activity dependent. So we can sometimes see quite enormous spikes of this in the clinic in patients who've done a lot of activity for it. It's an imperfect biomarker, but I would argue that sometimes it's imperfect till it's not imperfect anymore. And I'll point out one way we look at that, again, is to look at the sort of natural experiment. In this case, looking at patients who have, this was an effort to look at patients, what is the equivalent in the real world of people who carry mutations that are equivalent to exon 51 skipping outcomes. And you can see that many of these patients with certain versions of the equivalent genotypes are actually asymptomatic, including normal CK. So I'll point out at deletions of exon 50 to 51, 40% of the patients in our database, in fact, had normal CK. In fact, because this is in a hotspot of where deletions occur, the absence of a large number of patients with that mutation suggests to us that some of them don't even make it into these databases because their CKs are so normal, the diagnostic biomarker. But we can say that we can predict that certain genotypes are associated with relatively mild outcomes, and we should keep that in mind. A more dramatic example of this is actually from exon 45 skipping. So these are from observations in a database of at this time, about 1,200 patients. All of the ones that had the same outcome potentially as exon 45 skippable patients. This is also in the hotspot region for deletions. We observed that 2 of the genotypes in there, 45 to 47 deletions, 45 to 48 deletions, are Becker patients who no one lost ambulation before age 40. And we observed 4 cases of 45 to 46 deletions also in frame who had typical DMD. So they have a non-functional version of the protein. But even at this time we put in this paper, we didn't observe any 44 to 45 deletions. We were surprised by the absence of those patients and predicted that they were not in there because they would not have shown up in the database because it was a functional version of the gene. In fact, there's data, now I'm showing data that's publicly available from Avidity. I'm not representing Avidity here. This was publicly available on their website earlier, but kind of striking support of this idea. So here's a case where Avidity, which uses an IgG fragment to conjugate to PMOs to get them actively uptake into muscle, get active uptake into muscle, and does so in preclinical models quite strikingly. In a clinical trial of this, patients treated with PMOs show significant expression of dystrophin, as you can see in the 5 and 10 milligram per kilogram group, at levels of about 30% of normal, but perhaps more strikingly show a really profound correction of serum CK, including patients who have complete normalization of serum CK. So I would argue that CK is not a great biomarker until we see complete normalization, in which case we'd no longer have a friable muscle membrane. So in this case, it's a very valuable one if we return that to normal. A corollary to this is we are now increasingly finding in our large genotype-phenotype database where we look at this, an increasing number of patients where patients being identified or people being identified with deletions, large in-frame deletions in the gene by preconception testing or prenatal testing who have no symptoms whatsoever and family members who carry the same mutation. So the range of what we accept as the dystrophin variation as a gene, as a protein, is much wider than we understood because all of the databases of variants in the gene, the entry to all of them was dysfunction of some type or another. So I'll close just to say another reason that we don't want to rely too much on numbers, with one more case example of a young man who had a putative nonsense mutation, should make no dystrophin whatsoever, in exon 42 of the gene. Turns out if you splice out exon 42, you maintain an open reading frame. When he came to us, it was quite clear he looked distinct from standard Duchenne dystrophy. We did a fairly detailed analysis, and it's very hard to see from the back. The upshot of this is it alters the splicing pattern. He has levels of about 6 to 10% of mRNA that lack exon 42 with a nonsense mutation, and he makes levels of about 3%. And that's good enough at 3%. Here at age 16, he was going out and pinch hitting. He's a fanatic for the Cleveland Guardians, and when he goes out and pinch hitting and is walking around the campus of Ohio State University now. So we have to be very cautious in correlating amounts to function. We have to think about this on a genotype-specific level. So in the case of DMD, the caution about this is a biomarker is the degree of membrane protection conferred by a therapeutic dystrophin is not only dependent on the amount, but also on the functionality of this therapeutic dystrophin isoform. So comparing post-treatment levels of microdystrophin to normal dystrophin is challenging, will be challenging. I'll add just on the clinical side, in the presence of uncommon or novel genotypes, we probably want to be cautious in recommending microdystrophin therapies only after we really want to know what's the known or associated genotype or phenotype with a given unusual genotype. And then exon skipping approaches, including vectorized exon therapies, may lead to full-length or full-length equivalent dystrophins, in which case the dystrophin biomarker pattern will be different as well. So I'll close just with acknowledging many people who's glancingly related to the data I showed today. Thank you very much. Happy to answer any questions.
Thank you, Dr. Flanagan. I think we have time for maybe one question.
Hi. Rachel Salzman Armitage. Great talk, Kevin. Thank you. On the clinical trial with the N of 3 that you showed us briefly, it looked like the data on the infant were quite distinct from the older children. Can you just reflect on that? When I think about an infant, obviously they're younger, and then I assume the phenotype was different, and then why such different outcomes on the clinical trial?
Right, it's a little unfair to flash that by without the whole story. We're not quite sure of the distinction. The vector biodistribution was similar at the first time point in all of the patients. It seemed maintained in the infant to a higher degree than in the other patients, but at time point 1, where skipping profiles were quite different, biodistribution was quite similar. We don't have a good explanation why. We're seeking funding to continue this and treat more infants and treat the safety profile will allow us to treat additional patients at a higher dose. So we would want to treat older patients at a higher dose. In the back of my mind, I wonder whether the fidelity of splicing is somewhat different. I'll also add that in the infants, we have never compared transcriptome, let's say, in a 1-year-old patient, a muscle transcriptome in a 1-year-old muscle versus the 8-year-old muscle.
Yeah.
Is there usually some altered splicing at the DMD? There's multiple DMD isoforms, actually, that might be spliced differently then as well. So we have lots of questions to answer about it.
Thank you.
Sure.
Thank you very much. I think this concludes our symposium. Thank you very much to all the attendees and our speakers for the excellent presentations. But do remind you to please fill out the survey in the HGCT app. It really helps to make better programming for you.
Yes. Thank you, everyone.
Thank you.